Welcome to this presentation on CH3 BioSystems, a protein methylation biology company. Our business application is dedicated to improving health and well-being through basic science research discovery. We all know good friends and dearly loved family members who had been vibrant and robust individuals for most of their lives until they were stricken by a debilitating and incurable disease. Whether it is a slowly progressive neurological disorder, a sudden cardiovascular complication, HIV/AIDS, or some form of cancer, the cure for all of these insidious diseases will only come from basic science research. Our customers are the tens of thousands of research labs and scientists from across our nation and throughout the world. These scientists are constantly seeking out new tools to understand, diagnose, and cure these illnesses. Over the last few years, protein methylation has emerged as a key process involved in all of these diseases. Our message for you today is that CH3 BioSystems has the resources and expertise to supply research and clinical labs with a revolutionary, patented tool set to help with the pursuit of discovery, including future expansion into the fields of biomarkers, diagnostic, clinical, and treatment applications. In doing so, we will not only promote scientific and clinical research, but will also create an entirely new market segment. To understand how CH3 products work, imagine a dark corridor with many doors opening into rooms that may contain important information about human disease mechanisms. Up until now, scientists would find one of these doors by accident, and they have suspected that there are other doors. We at CH3 BioSystems can illuminate these doors and give the scientists the keys to unlock them. CH3 BioSystems plans to enter the biotechnology marketplace in two stages. Initially, we will provide our molecular tool set to academic and industry laboratories engaged in research and discovery related to protein methylation in the biomedical sciences. Establishing a revenue stream and a high-profile identity in this first step will facilitate the second stage, development of diagnostic and clinical applications of the technology, which is especially relevant to diseases that are associated with methyl proteins. The future of the healthcare industry is evolving away from the empirical model of therapeutics to a more stratified or personalized form of medicine. The pharmaceutical giants are spending more and bringing less to the market. The era of blockbuster drugs may be fading in favor of a more individualized healthcare, which requires matching a patient's genetic and biochemical background with specific treatments. Biomarkers, defined by the FDA as objectively measured indicators of either normal biological processes, pathogenic processes, or pharmacological responses to therapeutic interventions, are expected to play a major role in the transition to personalized medicine. We at CH3 BioSystems believe that many chronic diseases exhibit important associations with methylated proteins that can serve as clinical biomarkers. The detection and quantification of these methyl proteins may aid physicians in the diagnosis and treatment of patients who present with characteristic patterns of protein methylation. Currently, there are 5 areas of medicine in which protein methylation has been found to be associated in some way with clinical diseases, including cancer and cardiovascular illnesses that affect very large numbers of people. The following slides list just a few examples of the hundreds of methyl proteins that have been identified. In this case, the methyl proteins that play a role in autoimmune diseases like scleroderma, lupus, and MS are shown. We are aware of at least 3 neurodevelopmental diseases in which methyl proteins are either the products of the disease gene or are involved in metabolic pathways affected by the pathological process. Proteins that are involved in the reproduction of viral particles, including those of the virus that causes AIDS, are known to be methyl proteins. Protein arginine methyltransferases, or PRMTs, are an important group of enzymes responsible for the addition of methyl groups to newly synthesized cellular proteins. Some of these enzymes, or the methyl protein produced by the enzyme, have been associated with certain forms of cancer, the metastatic spread of cancer, and DNA damage. Finally, in the process of normal aging, the breakdown of the many dozens of methyl proteins gives rise to a form of the amino acid, methyl arginine, that can inactivate a key metabolic enzyme in blood vessels. The inactivation of this enzyme is associated with increased risk of hypertension and hardening of the arteries. Even before biologists had compiled all of these examples of methylated disease proteins, it was clear to some that all of the methyl proteins that have been mentioned have a telltale signature of the site of methylation in their amino acid sequence. It is the simple triad glycine-arginine-glycine. Scientists at CH3 BioSystems realized that there was an opportunity to design simple peptides making use of this sequence to produce a patented antibody that can detect the presence, facilitate the identification, and determine the relative quantity of the methyl proteins present in biological fluids, tissues, and cells. In addition, CH3 BioSystems likewise engineered a second control antibody prepared in the same manner against a nearly identical peptide, the only difference being that there was no methylation present. This table illustrates that the methyl-specific antibody, anti-MRG, can be diluted more than 50 to 70,000 times, and it still recognizes and binds to the methylated peptide GMRG, but not to the non-methylated form of the peptide GRG. The opposite is true of the control antibody. Anti-RG binds to the non-methylated peptide avidly, but the methylated peptide is recognized very poorly. We can see these relationships visually on this experimental format known as a Western blot. First, in panel A, serum taken from rabbits prior to immunization with the peptide does not recognize or bind to any of the peptides applied to the Western blot membrane. Anti-RG binds only to GRG, and anti-MRG binds only to GMRG. In panel B, we see that the differential level of this specificity is 10 to 50 times based on the quantity of methylated and non-methylated peptides applied to the Western blot membrane. In panel C, we demonstrate that the non-methylated peptide can be converted to a methylated form and be detected by the anti-MRG. This is successful only in lane 5, where in addition to the non-methylated peptide, the enzyme PRMT1 and the methyl donor SAM are also required in combination. The recombinant form of PRMT1 and a stabilized formulation of SAM are also products that are supplied by CH3 BioSystems. Panel D illustrates a similar phenomenon using cellular extracts as the source of the methylating enzyme. This experiment illustrates the utility of the methyl-specific antibody in detecting methyl proteins in cells. In panel A, the methyl-specific antibody was tagged with a red fluorescent marker, so you see many large protein aggregates that bind the antibody, especially in the nuclei of the cells. In panel B, an antibody to nucleolin, the most highly methylated protein known, was applied with a green fluorescent marker. When the two images in A and B are superimposed in panel C, we find that the red and green markings have produced yellow profiles because of the co-labeling with both anti-MRG and anti-nucleolin, indicating that anti-MRG correctly identifies nucleolin as a methyl protein. Scientists at CH3 BioSystems are currently collaborating with an international team of pathologists and cell biologists to explore the utility of the anti-MRG antibody in detecting cancer-specific methyl proteins. Here we demonstrate that the state of protein methylation in two very different types of cells is altered during developmental changes in those cells. In panel A, proteins from the protozoan parasite Trypanosoma brucei, which causes African sleeping sickness, have been applied to a Western blot membrane. The procyclic form of the organism, PF, from the tsetse fly, exhibits a very different pattern of methyl proteins than that found in the human bloodstream form of the parasite BF. Very little reactivity of the control antibody, anti-RG, is present. In panel B, two novel methyl proteins are detected amongst the Western blotted proteins from cells stimulated to develop into neurons with nerve growth factor treatment. This experiment illustrates the detection and identification of one of the proteins that increases in methylation after stimulation with the nerve growth factor treatment. In this study, the investigators made an educated guess that since the protein HNRNPA1 was known to be a methyl protein, the degree of its methylation may change during neuronal development. The experiment was carried out in two stages. Stage 1 used anti-MRG to sequester all of the methyl proteins by a process called immunoprecipitation from non-treated cells or from cells treated with nerve growth factor for 5 minutes, 18 hours, or 20 days. Normal rabbit serum, NRS, was used as a control for any possible non-specific immunoprecipitation. In the second stage of the experiment, the immunoprecipitated proteins were applied to the Western blot membrane and reacted with anti-HNRNPA1 to determine if this protein was among the highly methylated proteins that had been sequestered by anti-MRG. Only in lane number 4, where cells have developed into postmitotic, fully differentiated neurons, do we find detectable HNRNPA1. Lanes 7 and 8 demonstrate that the starting amount of HNRNPA1 over the course of the 20 days of the experiment is equivalent, and the absence of HNRNPA1 reactivity in lane number 6 confirms that the results are not due to the nonspecific action of just any kind of antiserum. In this experiment, we overexpress the Fragile X mental retardation protein, FMRP, in cells, and these are illustrated in lanes 2 and 3 of the middle panel. FMRP is known to be a methyl protein, and furthermore, CH3 scientists discovered that nerve growth factor can activate PRMT1, which increases cellular protein methylation. When the Western blot membrane from the cells was treated with anti-MRG in the upper panel, it is clear that there is significantly more methylation of FMRP only in those cells treated with nerve growth factor, once again demonstrating the usefulness of the methyl-specific antibody for detecting changes in protein methylation. The last demonstration illustrates how the protein product of the disease gene responsible for Rett syndrome, known as MECP2, is methylated, and also how its methylation is also subject to regulation by nerve growth factor. The upper panel illustrates that synthetic MECP2 is recognized by anti-MRG only following incubation with PRMT1, the methylating enzyme. The lower panel once again illustrates by the two-step immunoprecipitation process that NGF treatment leads to increased methylation of MECP2, which is detectable with the anti-MRG antibody. In conclusion, it is clear that the field of protein methylation is growing as an area of basic science discovery with important relevance to the future of diagnostic and clinical medicine. The number of diseases that involve protein methylation and the identification of disease methyl proteins are growing with each passing year of research. CH3 BioSystems is committed to serving both the current and future needs of this important new field of life science.